Hi, I'm Skylar Sargent Walters. I am a junior at Brown studying computational biology, and this is Faces of Brown Research. The title of my project is Developing Deep Learning Methods to Catalyze Viral Discovery and Identification for Metagenomic Datasets, which basically means that I am building a new method based on deep learning to discover and identify highly divergent viruses from this very specific type of dataset called a metagenomic dataset. So deep learning is a really scary phrase, but it basically just means that we're doing a lot of linear algebra and derivatives to discover patterns in some input data. So my model, what it does is it will say, this sequence is what a filovirus looks like. This sequence is what an adenovirus looks like. And so the hope is that we can take those unidentified sequences from the metagenomic data, put it into the model, and it'll tell us, this is a toga virus, this is a filovirus. And then we can make identifications based off the model. So when you get your genome sequence for something like 23andMe, you just get your genome. But in the metagenomic sampling, we take your genome and everything else inside your body. So that could entail viruses, that could entail bacteria, in addition to your genome. And so often we take a metagenomic sample when we don't know what someone is sick with. So that way we could have this sort of stock of every single potential pathogen in your body. The issue is this type of data is really difficult to obtain clear viral identification from, just because viruses mutate really, really fast. And the sequences tend to be really short in length, so only about 100, 200 nucleotides. For reference, viruses tend to be tens of thousands of nucleotides long. So we really don't have a lot to work with. What I focus on in my research is building a new method based on deep learning to identify what those viruses are from this type of data set and hopefully be able to tell people what they're sick with, especially when we don't know. My research is pretty big in health equity because we take a lot of metagenomic samples when we don't know what someone is sick with, and this is particularly relevant to under-resourced areas with poor diagnostic tools. So ultimately, I plan to apply my model to this data set that my PI, Dr. Katie Siddle, has been working on. That includes about 500 cases from various parts of Senegal in which we don't know what people were sick with. For example, dengue virus, which is another topic of research at my lab, is a very geographically evolved virus. So there are very different strains from different parts of West Africa. And so I just think it's a really great opportunity for us to not just get closure for a lot of these patients, but also to identify and understand the viral landscape of a lot of these countries. I first conceptualized my project in November, and then I had the daunting task of finding funding for it. So I found the HIV and Emerging Infectious Disease Scholars Program on the Global Health Institute website. And I got really excited because it's for students who are doing the exact field of research that I am. It typically only funds medical students and preemies, but if you ask really nicely and you're an undergraduate, you might be able to get funding as well. So I applied to the program in January, and then I was given the funds to do my research this summer. They also provide really great basic translational research every week, so you get a lot of exposure to the medical field and topical research in MD, PhD related fields as well. I mean, I've always been into infectious diseases. When I was a kid, I was really into viruses in a weird way. And this is kind of my dream, to be able to do research in infectious diseases and infectious disease discovery. Even though I don't want to go into, medical school, I still want to be pursuing research in computation and infectious disease. So it gave me a really awesome opportunity to explore both those fields. I realized that I love computer science when I came to Brown. I kind of started taking it by accident because I thought that I was in genetics, and I was very much not in genetics. I was in CS17, which I love with all of my heart. And I just think it's a great opportunity for me to blend what I've learned in all of my courses, right? So I take what I've learned from genetics and I apply that to the extreme with working with so many different types of viral sequences. I created this project after taking deep learning with Ritamber Singh, who's also advised me on this project. I am able to use data compression methods that I learned in my class with Professor Huerta Sanchez. And I just think it's a really great way to blend everything that I have learned and create some project that culminates in this beautiful fusion of knowledge. I think it's really incredible to be living out Brown's open curriculum dream of exploring anything that I want to. I think that viruses are essentially just little computers. let's ignore the whole debate of if our virus is alive or not for a second, right? They are, at their core, a string of genetic code. And that code runs every time a virus invades a cell, every time a virus replicates. It is just running that code over and over again. And I think it's such an incredible opportunity to explore viruses as computers through computational tools. So I love the ability to be able to explore the genome in that way and be able to, search these little computers, something that makes them tick, that makes them so scary and infects people so well. So I'm just really excited to see where this research goes. So I'm only a junior, so I'm hoping to make this my senior thesis. I just presented at a conference called ABRCMS, where I received the presentation award for computational biology. And I just feel like there's so many things that I want to update in the project, right? I want to try adding new computational methods. I want to try adding more data because I can do that now. I want to try, integrating some sort of mechanism into it to make it better at finding unknown viruses. And I just feel like there are so many great resources here that I can use for that. I also want to thank my lab. Siddle is really great. Dr. Siddle is fantastic. And they're just so supportive in this whole virus discovery journey. So it's really awesome.